Hello, everyone. Welcome to the fifth Open Science Training Series. We're very delighted to have Justine today with us. And Justine is in the final year of her PhD. She works in the Network Neuroscience lab studying with Dr. Misic at the Montreal Neurological Institute. Her research is focused on how the brain operates as an integrated whole. We'll present the data science era of neuroscience. Welcome, Justine.
Thank you. Thank you. Hello. It's really great to be here. I've never been here before, and this room is like already very cool. Like there's two fireplaces, very nice. Let me know if I should like project more. Also, let me know if I should speak slower. I tend to speak really quickly, so I'm happy to please just yell at me if I'm going too fast. This is going to be a very kind of casual presentation. I'm very open to being interrupted. I'm very open for this being more discussion based. Also open if you have specific questions, we can talk about that. Really, I'm just going to be telling you about my experience with open science and open data, and hopefully that helps you. And probably there will be things that are more specific to you that I don't know as much about, but I'm still happy to kind of wonder aloud. Yeah, so okay, so let's get started. So I've decided to call this the data science era of neuroscience because I've been thinking recently in general about how we have this kind of new way of doing science that was not really an option like 50 years ago. I think what we learned in elementary school and probably how science has like been done for a long time is that you have a specific question and then you kind of design a specific experiment to get at testing that hypothesis, and then that's your results. That's what you look at. But now I feel like there's this time where all these people have data that they've been collecting and it's becoming more and more open and accessible. And so as a result, you can start to ask questions that don't necessarily require you to design an experimental kind of setup. Of course, that means you're restricted in some ways. You can't necessarily do things that are very specific to your problem. You have to be like a little bit more general with the types of questions that you want to ask. But in any case, it's kind of like a new way of doing things. Data is plenty, right? So over the past, let's say 20 years, people have been collecting data, and I'm not saying that it's open necessarily, but there's all this data sitting in all these labs in some hard drive somewhere or somebody's computer. And probably a lot of it is actually relatively accessible. Obviously, there's ethical constraints with like individualized data or specific patient populations or whatnot, but a lot of it is just people collected data and didn't think to like make it open. Or also the fact that it's only until recently that we have things like the internet. We can like share things. And so I think there's like more and more people are thinking about accessing data all over the place, but also broadening our horizons a little bit so that we're not just limited to the one thing that we like to focus on or the one thing that we know really well, but also being able to like make bridges with other scientific disciplines. And so as a result, I think science is becoming more and more of a team science, right? So it's less of like, it's just me or it's just my lab and we're going to do this one very specific question. And I think it's becoming more and more, I'm interested in this, but this other person knows about this other thing. And I would love to make a link with that. So I'm just going to shoot them an email and see if we can make that happen. I think that's becoming more and more of a thing. Okay, so let me just like preface this with like, why am I here? Why am I here? I think so pretty much in 2022, I won this Open Science Award. And ever since that happened, I think people are like, I'm an open science person. And then I was like, oh, okay, I guess I'm an open science person. Like now people think I'm an open science person. So I kind of like, which is good for me, right? Because like it's like when you're asked to teach something and that's when you really like learn it because people expect you to know your stuff. You're like, guess I better know my stuff now. The whole thing was like really an accident. I didn't go into it thinking I'm going to be this open science hero and do all these great open science things and like the world will be beautiful. I came in thinking I'm going to do my PhD, actually at that time master's, and I have some research questions, and then the whole open science thing just kind of like seeped itself in there by accident. And the reason I want to tell you this, I mean, you guys are already way ahead of me because you're going to an open science talk. Like that's like already miles ahead of what I was doing. But it's relatively like it's possible to let open science kind of seep into your research. And I want to emphasize that I'm still early in my open science journey, but also this like really obvious statement that you don't need to be an expert to get started. I think sometimes open science can kind of come across as though everybody's already like a genius at everything, like all these open things like using GitHub and like how to do this and how to do that. And it can also be, I mean, I don't know. I feel like open science people also love to complain. So it's always like, well, you didn't comment your code well enough. So why did you make it open if you're not even going to comment it? And like, yes, like, yeah, that would be great if you did everything right. Like, obviously, that'd be great. But also, if you just start with the baby steps, my first GitHub repo is an embarrassment. Okay. Like, it's an embarrassment. But it got me to the place where I was winning open science awards and then was like, okay, I guess I need to like figure stuff out and like actually comment my code and stuff. And like I think that can be okay too. Obviously, if you guys start as like 10 out of 10 students, that's great. You don't have to start as like me, but it's also okay to start kind of without really knowing your stuff. Okay, so what I'm going to talk about today, the first thing is it'll be sort of a personal kind of journey thing, what ended up happening for me, and a lot of it was kind of accidental. And so one of the things is how I found and used open data and integrated that into my work. Another thing is how I found and used closed data and that helped make that open. And then the third thing, which is literally just a single slide, is the importance of giving credit. Okay, part one, how I found and used open data in my work. Okay, so back in 2021, no, back in 2020, I started my PhD in 2020, and I was broadly interested in kind of integrating different levels of brain organization. So I came in with a scientific question. I was kind of wanted to know, if we have all these ideas about how brain function works in terms of like a distributed network, let's say, of like activity, and I'm thinking human brain here. Well, how does that relate to something like teeny tiny like gene expression, right? Those are such different scales. How do we kind of bridge that together? So I started with this sort of question, and I kind of realized, or I mean, also with the help of my lab, that there's so much data measuring so many different points along this hierarchy of spatial scale. So just to give you some examples, this is going to be a little bit specific to human imaging. So for those of you who are not doing human imaging, I apologize, but this is sort of what I know, which is for gene transcription, we have things like the Allen Human Brain Atlas. This was like this big, in 2012, this Nature paper came out, and they were doing spatial transcriptomics using microarrays across the whole human brain. And what's special about the Allen Human Brain Atlas, it's very spatially comprehensive compared to before. Usually people would take like a little tiny brain sample and be like, here's the gene expression profile. But what was special about the AHBA was that they took these profiles across the whole brain, and so you could get a brain map, right? How does it vary across the brain? And what's also special, it's kind of an aside, but if any of you guys are like collecting data and thinking about making it open, they collected MRIs, and so they had specific coordinates for each sample that they took. That was really important too, because people like us, if you say I took a sample in this like vague area, that's really difficult to work with. But if you say here was an XYZ coordinate, it kind of makes it easier to work with. So Allen Human Brain Atlas, great resource, great data set. BrainSpan, it's actually from the same group, but is looking at gene expression samples across the developing brain. Less spatially comprehensive, but more data points instead of just being like 6 brains that are adult. You have some brains that are from younger populations. And then this thing, Abogen, this is something that a previous PhD student in the group developed as a toolbox. It's super useful in accessing this data. So it makes it such that instead of being like super overwhelmed when you look at the website, you can download the whole thing with one line of Python code. It's gotten to the point where I'll see papers that refer to the Allen Human Brain Atlas as the Abogen data set, which is like not what it is. Like Ross did not collect any data. He just made it easier to work with the data. But obviously the people who work with the data, we think of it just from the perspective of the way we worked with it. So, I mean, maybe it's good to remember like where it actually came from and like, again, give credit to the people who like actually did the data collection. But in any case, so there's lots of resources for working with gene transcription. Neurotransmitter receptors and densities, there's different compilations of which, I also made one recently sort of recently, where we're looking at either PET or audio radiography imaging of different proteins across the brain. So there's access to this sort of information. Synapses, this is like, there is no like logo or nice thing, but UCBJ is a tracer that is there to map synapse density in the brain. So that's also something, it's a map that exists out there. It's open, you can kind of get your hands on it if you wanted to. Different cell types, there's different ways of approaching this. Some people actually just think of cell types as an average of specific gene expression markers. So it's not actually a measurement of the cell itself. It's like, these genes are only expressed in inhibitory neurons, so let's take an average of the spatial expression of those. There's a bit of an assumption there that like gene expression is correlated to the actual expression of those cells. But in any case, the Big Brain project, I don't know if you guys know, but it's this big project in a collaboration between the Montreal Neurological Institute and this group in Jülich in Germany. And pretty much they took a single brain and they sliced it up in these 20 micron thick slices, and then they stained it for the cell bodies, and they imaged it and reconstructed it in this 3D brain where you can literally just like it's like you're in a video game. You can kind of zoom in around the brain, zoom into different brain regions, and just take a look at the cells that are there. And from that information, and people have already done this, so you don't have to do that, but from this information, people can derive things like cell densities of different brain regions. How many cells are there? What is the distribution of the cells? Because we can start to differentiate layers, things like that. And then, of course, brain structure and function, there are these huge data sets like the Human Connectome Project, collecting lots of, this is mostly MRI now. And then also for function, I mean, there's fMRI, which counts, but I think with function, there's also a lot of like interesting meta-analytic tools that are coming out. So instead of purely open data, as in I collected some data and then here's the data you can work with, there's this idea of the paper itself being the data. So for example, if a bunch of people report this brain region was activated when we did a working memory task, then the meta-analytic tool would tell you something like which brain regions are more related to this appearance of somebody in a paper saying working memory task. Does that make sense? So it's not like somebody went in and collected, I'm going to get a brain map that represents working memory. They are looking over, I mean, I'm talking right now specifically about neuroscience. Can you guys see my mouse? Yeah. Here. What it's doing under the hood is it's saying across, let's say, 15,000 different papers, fMRI papers, we're going to look at the coordinates that they listed in the paper. We're going to look at the words that they mention a lot. And we're going to just make little matching of, let's say they use the word or the phrase working memory more than like 1 in 1,000 times, and these coordinates keep popping up. That's a strong association. So that area in the brain seems to always be reported with the words working memory. So you see how it's sort of it's meta, right? It's like open data based on open papers, based on probably closed data, honestly. Like all those fMRI studies aren't necessarily open. In any case, so here's a bunch of data that I was sort of aware about or kind of like dug into and realized that there are all these things at my fingertips that are available to me if I wanted to start to play with this. And the question that I came into my very, very first project, it was broadly about mapping different levels of gene expression in the brain to different functional or cognitive specialization. So this is a very like typical data science project. It's kind of big data in a way. You have this big matrix of brain regions by genes. This is like 20,000 genes. Then you have this other big matrix of brain regions by different, I'm calling them terms, but it's like any sort of cognitive process like working memory, or it could also be a behavior like sleep or eating or whatever. Now the thing is, what's the data? Like, what am I actually working with here? And all of this data was open. So I came into this like I didn't like email anybody. I didn't ask for anything. I was just like, okay, let's like see what's out there. So Allen Institute, I already talked about, that's the gene expression data. And again, that because of Abogen, that toolbox that I mentioned, which huge caveat, the person who like developed it was a PhD student in my labs, right? So that's a huge resource, or I already kind of had, and the whole thing's a bit serendipitous, right? Like I went into this direction probably because I was in an environment that happened to have these resources. But yeah, so I could pretty easily get this region by gene expression matrix, and trust me, you guys can also easily get the gene expression. Like it's really one line of code and it'll download it for you. And then on the other side, this is the thing I was talking about, neuroscience, these probabilistic activation maps of different terms. And then after that, it's just a matter of a bunch of data science skills. So I think when it comes to open data, a lot of graduate students, there's just like new phenotype that's emerging of people like me and maybe like you who are really becoming more and more familiar with data science type skills rather than necessarily, like for example, I've worked with plenty of fMRI data, but I've never collected fMRI data. So I really don't know much about the details. I mean, besides what I've read or like what I kind of know on a cognitive level, but I've never really like handled it. You know what I mean? Like I haven't had to like deal with all the annoying things that I'm sure that people who collect fMRI data have to deal with. And so that's like on one hand, a lack in my knowledge. But then what I have on the other hand to kind of counterbalance that is more an understanding of these data science skills, being able to integrate things. And so things like that would be organizing and cleaning data. By the way, ChatGPT makes this so much easier. Like you guys, this is a good time. This is a good time. Like now, I mean, obviously be careful. Don't do anything stupid. But like it's much easier to wrangle data now, I think. A lot of learning how to use techniques that can integrate big data sets, things like machine learning methods or statistical methods. A lot of correlations, because here's the thing, when you're doing something that relies on open data, you probably don't have tons of control over the experiment, right? So for example, gene expression and these kind of meta-analytic patterns of cognitive activation, these are both normative, healthy control, different populations, and we're kind of sort of hoping that it's representative of the population. It's not going to be like a very specific condition or a very specific demographic, or even a very specific perturbation, right? Because I don't have control over that. So there's going to be things like a lot of correlations, lots of associations that probably aren't causal. And then like the one thing I really want to emphasize in general when working with open data, but also when approaching neuroscience with this data science view is that, yeah, go ahead. No, sorry, you're right. I like jumped that. I was okay. The end of my sentence is just that like plotting is king. Like always look at your data. Always look at your data. You get a correlation, don't just report the correlation coefficient, look at the correlation. And that's like hugely, hugely helpful. Anyways, yeah, go ahead.
I mean, sense of the thing that we can make it.
Yes, please do. Yeah.
I mean, isn't there like a real danger, like not having the domain expertise? So, of course, that correlations and plotting is super important, but without like a core domain expertise, like, like somebody who doesn't understand expression, like gene expression, like, I mean, you could be way, way off, and you wouldn't even be able to pick that up without somebody who has the appropriate domain. I mean, that just, it's the same with ChatGPT, right? I mean, you can use it for stuff, but if you don't have that basic knowledge, I mean, it might just not to the ton of garbage, right? So I think isn't that really dangerous that
Yes, that is, I'm really glad you brought that up. That is hugely important. I think so at the very beginning when I said this, like science becoming more and more team science, the one strategy I use and I highly recommend for anybody is that when you start to dive into these things where you're working with things that you don't necessarily have the domain expertise in, that you always include somebody who is a domain expert. You always have, so for example, when I work with gene expression data, there's always somebody that knows the genetics inside out, so they can tell me this is not, I mean, obviously do your like due diligence, you read and you try to understand what you're doing and you try to be careful about what you do. But that's why these papers are starting to get so many authors, right? Because when I work with, for example, neurotransmitter receptors, I'll talk about it a bit later, but I had this project where I was collating all these group average maps of PET images, and I hadn't worked with PET before. And so there's all these people, essentially what I call like the PET gurus who are there, and like, they could tell me, this is what you need to know. If I had an analysis where I said, hey, I see this association, does this make sense to you? Or do you think that this could be due to some sort of artifact in the PET imaging that I don't know about because I don't work with this data? So yes, domain expertise is definitely really important. And the way I think we get at this when we go further and further away from a very focused question is by bringing in more and more people. So you have all these experts kind of on your panel that you can kind of wrangle with this project together. Does that make sense? Yeah. But yes, that's a really good point. So here in this project, I sort of have just like some advice, I guess, when I'm working with open data, like how to sort of manage it. And here's this other example of the brainspan data set I also mentioned before. I wanted to replicate what I found in an independent data set and also that would look across time. And this is another thing maybe to get at the domain expertise thing of like, if you can work with open data, then you can probably replicate it in another data set, and that's also really valuable. You get a sense of how much do you trust what it is that you're doing. And for this data set, I just kind of want to make a little point that if you know somebody that has already worked with it, is very intimate with the data set, why not enter into a collaboration with them? Like get them to share their knowledge, right? We don't have to keep reinventing the wheel and trying to, if they already realize the sticky places where you might fall into kind of making an error, it's good to like get that from them, to learn that from them. Okay, so part two, how I found and used closed data, aka how to send an email, and that's like really like pretty much what it is. But what happened, so I did that first project. I'm not really going to tell you guys about the science because we're more here to talk about like the open data, the open science element of it. But I did that first project, and then I was sort of thinking, okay, so gene expression is cognitive, yeah, okay, but that's like really hard to think about conceptually because like what are all the steps in between? And something that was much more obvious to me or much more tangible, I guess, are the neurotransmitter receptors and transporters that are expressed at different synapses. Because of course, depending on, I think of them as ears, right? Depending on the ears that are listening for the signal, that's going to have a huge impact on the kinds of dynamics that you see in different brain regions and also the kinds of connections that you see between brain regions. So in any case, that was a scientific question. So this is another example of me coming in with a research question. It was a question for the sake of my PhD or for the sake of my, I guess, curiosity. And then it sort of accidentally became like a whole open science venture as well because I needed data to work with and I didn't have PET data myself. And so I contacted some people that I knew that had been working with healthy control maps of different receptors and transporters. And I said, here's like mGlorified. This is from the Douglas actually and McGill. I said, would you be willing to share with me your group average map? Group average because there were all these kind of constraints about sharing individual data. And I was kind of in a lucky situation where I was okay with just looking at the group averages. But so I asked group average, template space, brain maps. And I was really surprised by, in general, how positive people's responses were because I thought beforehand that people would be really kind of, they've collected this data and it's took a lot of time and money and probably they want to keep to themselves. And so I thought, I don't know, I went in like ready to really like beg really hard, I guess. And they were like, "Oh, great. Yeah, you should take it. Go ahead." And I think people like sometimes forget people are just like really excited. We're excited about the thing they're excited about, you know? So they were like, "Well, I've collected this data anyway, and I've kind of already done the stuff I want to do with it, and I'm very happy if you are interested in like trying some other things." So I think this is in general something that people underestimate, just the power of asking. Either something you want, see if you can ask for it. In any case, here's how this blew up. So it started with, here's a group at McGill and Douglas, people we kind of knew, and we've got this group average map of N-GLUR5, one of the glutamate receptors. And then there were some other people at UQAM, so this is still Montreal. And then Western, so we went a little bit like outside of Quebec into Ontario, and then to Ottawa too. And then eventually there was this like pet center in Turku that helped us out a lot. This actually was linked through one of the people, Lowry, is he there? Yeah, Lowry Tuominen. He is currently a PI at Ottawa, but he is Finnish and did his PhD in Turku. And he's a pet guy. And so he said, "Oh, well, let me get my PhD advisor into this. He leads the pet center and he would be like really happy to contribute, I think." And so that's how we got this link to the Finnish kind of community, pet community over there. That brought us down to Yale, where we also had another Rick Carson at that time. I don't think anymore, but at that time he was the head of the pet center there. So we got a bunch more receptor maps. And then it really exploded. At some point, I think I had the preprint out, like I had the project was done. I put it out on bioRxiv. I think my PI tweeted about it back when Twitter was like normal. And somebody commented like, "Hey, I have an NMDA receptor. Are you interested?" And I was like, "What? Yeah." Like I thought I had to beg you guys. You guys are asking me. So then we got like more data because other people sort of just saw our work. And here you can see kind of how much it expanded. It was quite global. A lot of people shared these group average maps. And what blows my mind with this whole project is that all the data was always there. It's just been sitting there. You know what I mean? It's been sitting there. And I did not do any of the hard work of like actually collecting this data. All I did was send an email. I was like, "Would you guys share with me the group average?" And don't get me wrong, it took time. Like it takes time and like a lot of emailing and whatever. Like it's a thing. But it was, I think, like hugely valuable now to have all these receptor density maps in the brain, because this is of course such a like important feature of brain signaling. And I was like, how has nobody else thought about, anyways, whatever. So just to say, like, if there's something that you'd like to study or like to relate your data with or your analyses with, not just human imaging, I just mean in general, if somebody has already worked with that somewhere, it does not hurt to send them an email and see if they would like to share, and of course enter into a collaboration with you. So yeah, in the end, that ended up being this big neurotransmitter receptor atlas. I put all the data, I put it on GitHub because back then I didn't really know that there's like other better places to put data. GitHub is obviously mostly for code and not for data, but see, I was like naive. So I was like, well, I mean, it's part of my project. So I kind of had it all organized in one way. It was on GitHub at the time. And then that sort of spun off. So another example of how things like, I felt like open science was like a being. It was just like constantly like hammering me with stuff because first this like pet thing happened with all the receptor atlases. And then there was this like other open science venture of turning that into a toolbox more broadly. And that of course is also a huge open science kind of endeavor. And so that sort of brings me into my next, so I don't know if you remember, but I did part 1 and then I did part 2. And this is part 1.2 because it's technically about open data, not closed data, but it's comes off of part 2 better. So like, whatever, it doesn't matter. I hope you're not, don't pay attention to the numbers. So at this point I was like, okay, I guess I'm an open science person now. I kind of collected this or created this atlas that I made openly available, obviously with consent, right? Like when I asked people to share their data, I was like, "Are you okay with me putting this on GitHub, making it open?" And people were very positive about this. And then we thought, okay, well, it seems like people are really excited to be able to relate the feature or the phenotype that they study to all these different scales of brain description, from the micro scales, from things like gene expression, receptor densities, cell types, whatever, all the way to the macro scale, things that you'd get from neuroscience or whatnot. So we thought, what if we were to integrate all of that? And just to give you a little conceptual sense of what the idea behind neuromaps was, which to kind of jump the gun is going to end up being, excuse me, going to end up being a toolbox. Imagine you have this like map of the brain, like a globe, and you have a sense of where the continents are, and that's great. And let's say you also know the connections between these different kind of regions of the earth. Obviously, this is a metaphor for the brain, but you know what I'm saying. You might be interested in some feature, let's say energy consumption. So maybe you're interested in a map of where energy consumption is the most, or maybe you're interested in some other feature like these are supposed to be cell towers. So like where different cell towers are and where you have good signal, where you have 5G signal. But when we start to layer all these maps on top of one another, we get a sense of how they're all related together. Like they're not independent, right? I mean, sometimes I think when we do neuroscience, especially very focused neuroscience, which don't get me wrong, is very important, we sort of forget that the thing that we measured is related to literally everything else. And so sometimes it can be helpful to remind ourselves, especially when we have access to these other maps, that they're all interrelated. And then you can throw other things like force density or like river systems or whatever. And then we just, in general, get a more global understanding of organization, but also probably function, right? Like where on the Earth, let's say, are regions that are more populated or whatnot. And so what do we need to do this kind of large scale characterization in the brain? We would need all the maps, right? So library brain maps. You'd need a method to compare them. It's one thing to make qualitative comparisons and just look at them and say, yeah, they kind of look similar. It's another thing to make quantitative comparisons. And you need to be able to sort of contextualize the map that you get with the broader literature. And so that's kind of like what this helps out with. So what NeuroMaps wanted to do was it sort of provides a suite of transformations. So this is to help with the quantitative comparison. If you're not doing brain imaging, don't worry about it. But essentially, it's like the analogy I give to my mom when she's like, what are you doing? Is that I say, it's like if I gave you a globe of something, like let's say Fox density, and then like a rolled up piece of paper of a map that represents like force density. I asked you to compare them. You could do that qualitatively because you could sort of, the anatomical landmarks, the geographic landmarks, and so you can make that comparison. But to do that quantitatively, you'd need them to be in the same space. So you'd have to convert the piece of paper to the globe or the globe to the piece of paper to actually run something like, let's say, a correlation. So that's what the transformations are for. And then the really important thing is the library, right? Like having access to all these different phenotypes, being able to statistically compare them in a rigorous method. I'm not going to go into that very much. But ultimately, to be able to do some sort of enrichment, get a sense of if I have a feature, what else is it related to? And here's maybe the more relevant part for you of all the data that's out there, just to give you a sense of here is a bunch of different features of the brain. And again, this is human, not just imaging, but this is human. So I don't know how much this would extend to other species, but there's so many different measurements that exist that are surely all related to one another, and they're openly available. That's the main thing, right? They're all open. And so if you had a brain map and you wanted to contextualize it, you really can. Like you really do have the access to all these different, not just the data, but also the tools to quantitatively compare them with one another. And yes, Abidjan again. So here's a few like pieces of advice about this kind of work, working with data that was previously closed and eventually you helped make it open. So one of the things is just send that email, like ask, but be specific about what it is you're asking. And for me, what I did when there's probably many ways to make it work, but what I always did was I knew what I was asking for. And I had a bit of a like slide deck. I had some presentation, like a bit of a presentation to show here's what I'm going to do with it. So here are the kinds of analyses I'm going to do. I had some preliminary analyses already. So they had a sense of like, oh yeah, this is like a legitimate project. This is going to be something, and I can understand where I fit into this as a collaborator. I think that helps a lot. I strongly recommend, I don't know, I guess like it's different for different cultures, like scientific cultures in terms of like co-authorship and whatnot, but it has helped me immensely. Like one of my favorite parts of doing science is working with the people who are the experts, because I sort of get to sit there and soak in all the insane knowledge that they have about this very specific thing. And then hopefully I get to sort of relate that to the other pieces of incredible knowledge that I'm getting from some other person, right? So for example, I was working with brainstem data and I didn't know that much necessarily about the brainstem besides, I guess, what you learn in like Neuro 101. And just all the wealth of knowledge of this person who understands the functional neuroanatomy, but also the imaging of the brainstem so well. But then I kind of sat with all that information, and I had already been working with the neurotransmitter receptors and transporters. And of course, as you guys probably know, a lot of these neurotransmitters are synthesized in these brainstem nuclei. So I was making all these links that if you're super focused in one domain, you might not be able to make those links because you haven't kind of zoomed out. I mean, it's a trade-off, right? Like I like to joke that I'm an expert in nothing, because I'm not. I'm just good at being friends with all the experts. So this is sort of like that's the strategy, I guess. But yeah, so I think working with these experts has benefited me hugely. And I hope it's benefited them too. Make sure that you give proper credit to the person who collected the data. This is really important. Sometimes, like the whole thing about the Abidjan data set, Ross was not the one who collected the gene expression. That was a huge amount of work. And so the person that you should be citing or giving credit to is the people who collected the data. And then if you use a tool, that's great. You can also give credit to that too. But of course, if you're using the receptor data that I made open, I'm not the one that collected that. All I did was send some emails and like put it on GitHub, right? So the people to be crediting are the people who actually did the work to collect the data. And I think it's important to be like really clear from the get-go what you're planning to do with the data, how you're planning to use it, and then if you're planning to make it open, what that means. And so in my case, it was like, I'm going to have these group average maps. They're going to go on GitHub. That means that anybody could use it. I'm going to try my best to make it really clear to other people that they should be citing you, not me, right? That's kind of important. And it doesn't always happen. I think people don't always really, they think of the data, they see my repo name, and so they just cite me. But then there's all this like text of like, here are the people you should be citing if you use this data. And I think people are like maybe I'm just like too naive, but I think people are really, or can be really positive about sharing their data and seeing the kind of impact that can have when people that have different domain expertise can use the data that they collected. This is very kumbaya, but anyways, yeah, you get my point. So this is like a single slide, my last slide, just to really emphasize credit where credit is due. When we use open data, there's a lot of work that went into that open data. Please give credit to the people who do the collecting. So yeah, this is my like spiel. I'm happy to talk more about here, like my thanks slide to you guys, the organizers of my lab. Obviously, after saying all that, I better say this too. I'm happy to answer any specific questions if there are things that are more like tailored to you or to tell you more if you want to know something specific about like, I don't know, how I did something or other. Yeah, let me know.
Yeah. I don't mean to talk, but
No, please do.
I mean, I have a question, like giving credit where credit is due. I think that's such a great conceptual idea, but we live in a world where the first orchestra, the last author, it matters, we get jobs. And I just came up with very specific. I work with the industry too. Fantastic paper is meta-analysis done by very, very some research science. He's in London. And then this paper has just made a huge impact, but he collected the data from like 25 different labs, including the one from that we did here in academics. I mean, and it just goes back to the question. I mean, because if you want to say, hypothetically, get a faculty position, you need to be that author, but then you have collected all of that data. And especially now in our current world, like the Canadian government is a huge amount of over 17 or 18 years, this was all taxpayer dollars, right? So like where did it benefit Canada? Like and so these are like huge questions, right? With open science. And then it sort of leads to my second question, which is all of this happens, say now we have the backdrop of OpenAI. I mean OpenAI, it's like the foundation of all of that work was done right here in Montreal with Joshua Benji and in Canada. But Canada is nowhere on the scene. Like we have all of this company, it's like hugely profitable. So how do we get from conceptual idea that we should share, we should benefit collectively? I mean, and yet make sure that we have that. So I think that is an important point. I mean, all of this work that was done, that was all funded by CIHR. Over 18 years. It's a lot of money from taxpayers. Eventually, I mean, the taxpayers should derive some benefit from it. This is my point. I mean,
yeah,
how do we address that?
That's a great point. And to be honest, I don't think I've have anything super intelligent to say, but yeah, I guess I don't think about it very much, but from that perspective, as somebody who's like, I can see it being much closer to home for the people who are the data collectors. And yeah, I haven't thought so much about the national perspective of like, hey, these are Canadian taxpayer dollars, like where's the Canadian benefit? What do you think about like, if this has a huge impact to to the scientific community and hopefully to the ADHD community. Now, like, I don't want to come across as like, I mean, or I'm okay with coming across, I guess. Let me just throw it out there. I'm probably pretty naive when it comes to this subject, but doesn't that also sort of benefit Canadians? Like if it benefits, you know what I mean? Like if you kind of have open data and it ends up in this really impactful work about ADHD, let's say. And yes, so that first author in, I think in London, you said, you know, they get a lot of credit and maybe they get a faculty position that some of the Canadian didn't get. And that's a shame. But hopefully in terms of our understanding of treating ADHD, that's something that I hope is shared globally, would you say? As people who use open data, credit where credit is due. Yes, that's really important. And people who give open data, when it comes down to it, it's an extremely selfless act, right? I think people do that. And I hope that people who make data open realize that unfortunately the reality is that it probably means that they're not going to, you know, if you give open data, it's not like you're getting money out of that or anything. You know what I mean? Like you're not selling it by virtue of it being open. And so I don't know. Yeah, it's really selfless. Like I see a question in the back. Yeah.
Yeah, because well, thank you very much for your presentation and for your high level of enthusiasm towards open science. It's really appreciated. It's so I just want to comment and also and maybe you can discuss that a bit, but I do see like the challenges that comes with sharing data, you know, but at the same time. I think first that data does not belong to a researcher, and I think that there's this very old culture that we need to change. That you're as a researcher, you get funded to do a project. That data does not belong to a researcher to hide that data forever somewhere. I think, and if you ask, I guess many stakeholders, if you ask the perspective of participants, and say, would you be interested for your data to be used by 100 people across the world? I'm pretty sure most participants would be thrilled with that. Let's say that we can make the best, we can really squeeze out everything out of their data. So I think that there's really, I think there's a culture, and it's very understandable that many researchers wants to be protective of their data, but I think institutions like NIH and IMH still exist. I made it clear that all the data has to be pushed out there and be made available after 6 months. And I think there's been benefits of that. Like people just let's say in our world of brain imaging, schizophrenia, there's a mini consortium, and we keep seeing like new project, new ideas, that really still using the same data. And I think it's the field is that everybody can go at this open data. So I think we're not doing a disservice to those who created or generated that data. We give an opportunity to everybody, including folks who could never have the capacity or the resources to collect that kind of data. I think we're making research a bit more democratic. Having said that, you mentioned that it's all about emails, which I'm sure that's the case, but what happens after your email? Because like you need like data transfer agreement, like what is it seems maybe you could elaborate a bit of what happens after your email, after your yes, because I suspect there's some important steps that happen from making that data available, from that email to the access to it.
Yeah, that's great. A great question. Yeah, I love your first point about making data more democratic. I completely agree. I hope it becomes more and more the case. For what happens after the email? So to be totally honest with you, I think for people who want to share data that is more sensitive, which wasn't my case. So for data that's more sensitive, it's probably a lot more complex. There's probably and that's why I didn't get the individual brains, right? That's why I got the group averages. It probably is more something like data agreement and whatnot. But to be totally open, really, like, I swear, what happened was I sent the email. They said, sounds good. This is the person who's going to work on that. And then depending on the person, a couple days, maybe a couple months later, I got a nifty file sent back as an attachment of the, like, that was literally the level of the component. And, you know, I guess, yeah, sometimes I do think back and I'm like, wow, they really trusted me that I would stick to my word about, you know, I don't know, they were obviously included as co-authors. That was made very clear from the beginning, and that I would make this open the way I made it open and whatnot. But yeah, in my case, that's really all it was. But I imagine, especially if you're working with more sensitive data, it would get more complex, which I unfortunately just don't have the experience in. Yeah.
Thanks for the great talk. I think a big advantage of doing these kind of open data initiatives and combining data across different research studies is to kind of come up with new hypotheses and use the combination of these data sets as a hypothesis generating tool where you'd be able to correlate things that you wouldn't necessarily be able to look at before, and then take that information and go back to the people who generated it. I guess from your experience, have there been any hypotheses where after combining a bunch of data sets, you go back to the original people being like, I think I found something that's really related to your work. Maybe you should look more into this, perhaps experimentally. Like right now the direction that is most common is going from like individual or like more precise studies and then correlating them, but has it gone the other way around too, where you're taking it back to the people?
Yeah. Yeah, this is a great question. I also completely agree. It's very, I think people have I will get to your question. I'll try like an anecdote. I think people sometimes think that with all this open data that we're moving further and further away from experimental scientists, and suddenly we're just like living in an echo chamber of using open data and not talking to the experimental scientists. But I actually think the opposite is true, or I hope that the opposite is true for most people, that instead it actually really brings together people with different domain expertise into a lot more collaboration, a lot more communication, and exactly to just kind of loop thing of like, my work depends a lot on the experimental work, not just for the sake of the data, but for the sake of the knowledge, right? I have to come in like, what kind of questions do I even ask? And that's not coming out of the blue, right? It's coming from what people have done. For example, with the receptors, people did that at the level of a single channel, at the level of a single cell. That's the only reason I'm like, well, this seems reasonable to ask at the level of the whole brain. To get to your question, so I don't think I've had enough time, or not, I don't think, like there hasn't been enough time for me to actually see like an experiment come out of it, but definitely lots of ideas. So two things that come to mind, the brainstem data that I worked a lot with, I was essentially finding that a lot of this, properties that we see in the cortex that we usually think of as purely cortical, but a lot of the reason we think of it as purely cortical is because we're only looking at the cortex, or it's a little bit like redundant. I'm seeing a lot of stuff in the brainstem too, and so that I think has helped people think about like, oh, maybe I should go back to my cortical studies, but also the brainstem people saying, let me go back to my brainstem studies and see if I can also see these features emerge that I didn't previously think to look at. So that's one thing that was kind of fun. And another example that happened even more recently, as I've been working with different synapse types, synapse morphologies that are expressed. This is actually in the mouse brain. Different synapse morphologies expressed in the brain, and I was trying to relate that to different features of dynamics. So for example, if you have a very specific type of synapse, which maybe you guys already know this, but I did not know that there was like a million different types of synapses in the brain. I was really just thinking excitatory, inhibitory, modulatory, but there's like so much diversity. So if you have like a specific type of synapse, you're probably going to have a specific way of responding to the signal. And this is something that the experimentalists at the level of a single synapse, so we're talking like patch clamp experiments, they were noticing that, but I was wondering, can we see that? Can we relate synapse density to something like fMRI dynamics? Do we see specific features? And then I do see these specific relationships between elements of the time series, the dynamics, and the synapse morphology. And then the experimentalist goes like, oh, let me see if I can see those same dynamics at the level of that single synapse instead of at the level of the whole brain. Yeah, I mean, the experiment isn't actually done. This is just me saying, look at this cool thing. And they're like, oh, I should look into that. But as we know, we often say, I should look into that. And then like, don't. But yeah.
To the conversation around data sets versus like collected data sets. I think just like the scope of what we can accomplish with like merged data sets is just so much greater. Like a single like patient cohort in mouse, So I totally respect the value for like neuromaps and stuff like the data. So thank you for that. My question is related to like the kind of danger of open data that we were talking about earlier. And I think there's a trend now where a lot of imaging specifically, where there's a lot of pre-processed data are made available. And obviously, like as students, and it's a lot easier for us just to focus a lot more on pre-processed data. But especially in the context of like MRI, like our pre-processing choices right? So do you feel like there's any danger specifically in that aspect of the open science?
Yeah, I think one thing that I regret for sure in my own like kind of scientific career is that I always feel a little like an imposter from talking. For example, I was fMRI data in the brainstem. I feel a little silly telling about all these things and then not necessarily knowing I don't have the intimate knowledge of all the pre-processing that went into it. I do think there's a danger, and I think it's a shame that but at the same time, I don't know if there's like any way around it, that there's this kind of, I'm spending less time being intimately familiar with fMRI, but as a result, I can spend more time being able to link across scales, right? And so that's, I think, where the whole expertise things comes in, having the person who actually collected the data on the paper that you talk to, and hopefully they're really helpful and can let you know about how different things impact different things. I also think what I'd really love to see in the open science community in general, but it's just becoming more and more of a trend, is that these kind of projects that are, like you said, kind of broader in scope, but as a result, also much less specific, right? It's like sort of more general, like, oh, there's an association, but we don't necessarily know like the mechanism behind that. What I'm seeing more and more for that kind of work is that it's becoming commonplace that it comes with this supplementary section that's like 30 pages long and has redone the analysis in like every possible way under the sun. And I think that's, I mean, as much as it's so much work, I think it's a really good tradition and culture of, if I use open data, I'm also going to try to throw at it as many pipelines as I can and as many different parcellations, let's say, or different statistical methods. Whenever I make an arbitrary choice, I'm going to try to do it in other ways and make sure that I really can believe what it is I'm looking at. Yeah.
I also wanted to add that when you don't have the privilege of opening up your data, you can always open up your code for analyzing your data. You can nominize your data. You can just put out how you're going to analyze, like as in a pipeline or anything. And fully credit that as your work. And that really links to what is called open license that we're going to talk about on April 9th. To wrap up the talk, if we have any more questions on Zoom, we have to take them. No, the questions on Zoom? Last one.
Thank you so much. I mean, it's incredible work. Clearly, so much work went into this. It's amazing. I have a question about white matter. So, if I'm not mistaken, epigenes and neuromaps are gray matter only, unless I'm mistaken.
Yeah.
Is there any sources that look at this kind of across scale situation in white matter, or is there any like effort to maybe include certain tracks or even just like white matter skeleton into the existing toolboxes?
Yes, I think so. Any of the PET images are going to be brain wide. Sort of like, doesn't depend on where we are. If we're thinking like MRI in terms of different like specific diffusion sequence, like things like fractional I can do this. Anisotropy. There you go. Those kind of measurements. Right now I can't think of any that are fully open, but I have a feeling that it's either like going to be really soon, or it's the kind of thing where it just requires an email, and because people are collecting a lot of these and using a lot of these. And then the one person I would point you to, I'm sure you're already familiar, but Ted Satterthwaite at Penn, I think, is doing a lot of tracked work, right? Like Val Sidnor is like cortical thalamic, or thalamic cortical. I think Val Sidnor's are open, and I think that's more and more becoming a thing of trying to like get these tracks out there and the annotations on them. But it's true that in general, this multi-scale characterization is much slower than the gray matter, partially because like also biologically, you know what I mean? It's like this, yeah. Anyways, yeah, that's
Thank you.
nice.
All right,
Thank you.
Thanks for coming.